Genetics and Genomics in Medicine, Chapter 6, Principles of Gene Regulation and Epigenetics. Overview. In this module, we'll consider various processes that regulate how our genes are expressed, with an emphasis on the processes of transcription, post-transcriptional modification, and translation. Learning objectives. Following completion of this unit, students will be able to describe the basic regulation of gene expression, relate chromatin modification and epigenetic regulation to modifying gene expression, and explain underlying epigenetic abnormalities that are responsible for some common diseases. Module 1, Genetic Regulation of Gene Expression. To start, we need to understand two very important concepts: cis-acting and trans-acting gene regulation. Cis-acting gene regulation refers to specific RNA or DNA sequences that other trans-acting factors, which can be proteins or RNA molecules, bind to. So essentially, you have a specific sequence or element of DNA or RNA that is either close to or distant from a particular gene that is regulated, whose expression is regulated by the binding of a protein or an RNA molecule. Let's take a look at a picture, and that'll help us understand this more clearly. This is a slide that exemplifies various examples in nature of cis and transacting factors. In A, we show a chromosomal region, two homologous chromosomes. The blue one is the parental chromosome and the paternal one, and the pink one is the maternal chromosome. The rectangles A and B on each chromosome correspond to specific genes whose transcription is regulated, whose gene expression or transcription really at the transcriptional level is regulated by cis-acting elements or specific DNA sequences. And these DNA sequences are exemplified by the ovals and circles in orange. And so we see here that the cis sequences 2 and 3 regulate the gene expression of A, and the sequence 4 regulates the expression of B. When I say, in this particular instance, when I'm saying regulating expression, we're talking about at the level of transcription. We also see in that it can happen at a distance. So as I said earlier, these sequences don't necessarily have to be very close to the gene. They can be very distant, and that's what is shown by the sequence number 1, the oval 1. The hashtag symbol, that means that there's a great distance between 1 and the rest of the chromosomal region. B shows us how mutation or genetic variation in one of the cis-acting factors or DNA sequences can alter or disrupt gene expression. So that alteration could be upregulation or downregulation, but it's going to alter that gene expression, and that could happen anywhere that we have a cis-acting element. If we change the sequence, that's going to influence the binding of the transacting factor to that DNA element, because we have to have those two things working together, the transacting factor with the cis-acting element. The protein or the DNA is going to bind to that specific sequence of DNA. And then in C, we're actually showing how a protein that's produced on a gene that's located on a different chromosome, on a non-homologous chromosome, could influence the gene expression of another chromosome. So here we see the gene product Z, the protein that's produced on Z gene, is going and binding to the cis-acting element number 2, that in turn is influencing the gene expression of A. In addition to proteins influencing gene expression, cis-acting regulatory elements in mRNA are often located in untranslated regions, and those are referred to as UTRs. In this example, the expression of gene A is regulated by not only the transacting regulatory protein X, but also the microRNA Y that binds to the cis-acting elements in the 5' and 3' untranslated regions, respectively, of the mRNA that's produced off of the gene A. So the microRNAs are binding to the mRNA that's produced when the gene A is transcribed. So those untranslated regions are then modified. So it's not just the proteins that can bind, which we talked about in the previous slide, that can bind to cis-acting elements, but it's also microRNA. And we'll get into more detail about each of these examples in subsequent slides in this lecture. Section 6.1, Genetic Regulation of Gene Expression. Next, let's talk about two important concepts. The first is promoters, and the second of which are tissue-specific enhancers, silencers, and insulators. So that's what we'll talk about in this section, promoters, and in addition to promoters, tissue-specific enhancers and insulators. So when we talk about basal level transcription, we're talking about just the standard on-off of transcription of a particular gene. And it is promoters that regulate, that are the major on and off switches for this. Promoters can be characterized in several different ways, and first and foremost is that they're cis-acting regulatory sequences. So promoters, going back to our earlier definition of what a cis-acting regulatory sequence is a specific DNA sequence. Typically it's upstream of the transcription start site, and it's recognized, i.e. bound to, by the RNA polymerase. And think for a minute, which RNA polymerase would that be? And that would be the RNA polymerase 2 in concert with other general transcription factors. And remember, when we say factors, typically we mean proteins. Promoters typically have consensus sequences, and when we say consensus sequences, that means if we looked at all the promoters that were out there in nature, we would see some common DNA sequence features that are characteristic of promoters, and sometimes they're referred to as elements. And so in this particular slide we see that we're talking about the BRE element, the TATA box, the INR element, and the DPE element. This slide is showing you the specific sequences that correspond to the BRE element. So we see that the BRE element is typically either a GGG or CCA that precedes a CGCC sequence, and then the TATA box is a sequence that's rich in TATA. The reason that we see nucleotides one on top of each other is that because sometimes there's some flexibility in which nucleotide that's actually going to be. And then we have a similar example for the INR element and the DPE element. The numbers that we see, negative 41 to negative 35, and typically that's spoken as minus. So minus 41 to minus 35, minus 34 to minus 27 for the BRE and TATA boxes, and minus 2 to plus 5 for the INR, and plus 28 to plus 32. Those are where these promoter sequences are typically located with respect to the transcription start site. So that means that the BRE element is 41 to 35 nucleotides downstream of the transcription start site, and the INR would be anywhere from 2 to +5 with respect to the transcription start site. So you can see that sometimes the promoter can actually be upstream of the transcription start site. But at any rate, this is the promoter. Most importantly to remember is where the general transcription factor that initiates transcription and the RNA polymerase II will bind and initiate transcription of a particular gene. Now, what about modulating transcription? Modulating transcription is sort of like the fine tuning, so that's not going to start transcription like the promoter does, but there are other specific sequences to which other regulatory proteins can bind that will influence transcription and really modulate the higher, not just on, off, but higher, lower. It'll attenuate the transcription. And these are, again, are specific DNA sequences that are recognized in particular tissues, and that's one kind of feature that distinguishes them from promoters. So a particular gene for protein X, for example, might have the same promoter irrespective of what tissue it's in, but the enhancer, which makes that protein be produced more or less or transcribed more or less relative to another tissue, might be those sequences might be specific to a particular tissue. Therefore, they're not ubiquitous, and they're bound to by specific transcription factors. Okay, now let's just take a look at what some of these enhancers look like. Let's take, for example, remote enhancers. This is actually quite interesting. So let's consider the Ch06 gene, which is a 30 kilobase gene that is important in certain diseases, but it's actually regulated by a lens-specific and a retina-specific enhancer from the eye that occurs within introns of another gene. So you have this very large 260 kilobase region of a chromosome which contains the Ch06 gene, and then within another gene downstream of that are some enhancers that in turn are going to regulate the gene expression of the gene upstream of the gene that they occur in. So this is really quite an interesting feature. And so by definition, these are quite remote. So if they're quite remote, how do they actually physically affect transcription of that gene? Well, one of the ways that scientists have discovered is through the phenomenon of DNA looping, which is shown in this particular slide. We have the RNA polymerase on the right-hand side, and it's the polymerase complex that's sitting on the promoter for the gene that's shown by the blue rectangle. And then we have some proteins that are bound on an enhancer sequence, and the enhancer sequence is shown by the orange rectangle, and the green and purple circles are the enhancer proteins. And what happens is that the DNA actually loops over so that those proteins bound to the enhancer then come in close proximity of the RNA polymerase, and they can then influence, like stimulate, for example, the transcription. So this is really physically how this phenomena takes place. So it's important to know how these different transcription factors bind to DNA. So, what is important in that binding? Well, both the protein and the nucleic acid structures themselves contribute to transcription factor binding. The transcription factors themselves, which are proteins, bind to these short DNA sequences, and within the proteins themselves are specific regions called DNA binding domains. So not just any part of the protein will bind to this regulatory DNA sequences, specific regions, and they have names, and they have certain amino acid characteristics and also protein structures. One of the best known is a zinc finger protein, and another example is the leucine zipper. So that's important for you to know, and in a subsequent slide we'll actually show you how that protein structure facilitates binding to the regulatory DNA sequence. Other factors that influence transcription factor binding is the actual accessibility of the DNA binding domain itself on the chromatin of the DNA to be that harbors the gene that's to be expressed. So, as we recall from previous chapters, we know that DNA has a very compact structure. It's bound with nucleosomes into chromatin, and then there is localized DNA remodeling in the chromatin so that the DNA can be accessible. And so those factors that regulate chromatin remodeling will again influence the transcription. And oftentimes there are combinatorial effects. That means that multiple different transcription factors will work in concert by a binding to adjacent sequences to work together to alter gene expression. And so you have co-activators and co-enhancers and repressors working together to regulate the gene expression. On our next slide, we actually show you those protein structures in more detail of the DNA binding motif from the transcription factor that interacts as it interacts with the DNA. So in A, we're showing you the zinc finger motif, and it's called a zinc finger because it is a specific amino acid structure that almost chelates a zinc ion due to the presence of specific amino acids. And it's comprised of 2 cysteine residues and 2 histidine residues, and by virtue of their charge, that is how that they kind of encage this zinc molecule, or zinc ion rather. And in so doing, they form a unique structure that's shown on the right-hand side that interacts in a very specific way, and it forms like a finger-like claw that intercalates with the groove of the DNA double helix, and that's how that protein-DNA interaction takes place. On the other hand, we have the leucine zipper, and you can see that the leucine zipper again is forming a structure by interacting with another transcription factor. So one feature that I think it's important to note is that these transcription factors frequently bind DNA as a dimer, and that's because of the structure of the DNA double helix actually needs to have something binding as a dimer so that you can get coverage over the whole DNA binding domain. So this slide does a nice job of showing you the leucine zipper and the zinc finger. So regulation of RNA splicing. There are 3 fundamental cis-acting sequences that mediate RNA splicing. The splice donor, which is an invariant GU dinucleotide at the 5' end of the intron, and a splice acceptor, which is an invariant AG dinucleotide at the 3' end of the intron. And then lastly, there's a branch site that's close to the splice acceptor, and that also is comprised of an invariant nucleotide, and in this case it's an A nucleotide, and it's required for the initiation of splicing. And then, similar to what we saw with transcription itself, there are additional cis-activating elements called splice enhancers and suppressors. So we again see that in the instance of RNA splicing, just like we saw for transcription itself, there are specific cis-acting elements that regulate this process. And then here we show a picture that depicts the splice donor, the splice acceptor, and the branch sites. Remember, these are going to be adjacent or within the intron that's to be cleaved out, that's going to then allow for piecing together of the exons that actually will comprise the mature mRNA transcript. This picture depicts the additional regulatory sequences that are involved in splicing, and that includes those enhancers shown in orange and suppressors shown in black. So, when we're talking about regulatory sequences that are involved in splicing, whether they are the primary splicing splice donor acceptor branch sites, or whether they are the enhancer sequences for RNA splicing, one has to consider how is the splicing taking place. And so, much like we saw with transcription, the RNA splicing is mediated by ribonuclear protein complexes known as spliceosomes. And this is important for you to understand that it is actually groups of proteins and RNA that come together in this entity known as the spliceosome that effect the cleavage of the RNA and then the rejoining itself. So again, the spliceosome is a protein RNA complex that contains several types of small nuclear RNA, including U1 sRNA, which base pairs with the splice donor sequence, and U2 sRNA, which base pairs with the branch site sequence. And it's important to understand the genetic control of splicing because mutations that cause abnormal RNA splicing are a relatively common cause of disease. Another important concept for us to become familiar with is the notion of alternative splicing. So it's reasonable to understand that if we have a given gene that's made up of several exons and introns, and depending on how those are spliced and then combined together to form the final mature transcript that is translated, could lead to different gene products. And this particular slide shows a variety of ways that this could happen. And in A, we see the intron sequence, which is shown in gray, is either excluded from a transcript or retained. So you have the exons shown in blue rectangle, and the intron is shown in gray. And in the simplest example, this would be spliced out. This intron would be spliced out, and you would end up with the blue rectangle comprised of exon 1 and exon 2, although they're not labeled 1 and 2 in this example. Alternative splicing goes to show that, for example, in B, we would have alternative splice donor sites, B, or alternative donor sites, C, where you would then splice at a different location. So if it was to happen in B, then the amount of exon sequence from exon 1 would be less, that would be joined with exon 2, and it's vice versa in the example in C. And in D, we show that the exon there shown in green could be included or skipped altogether. And so that is another example of exon skipping. So when you have multiple exons, and sometimes genes are quite large and may have 50 or more exons, and so in different tissues, we can have expression of different combinations of exons through the phenomenon known as alternative splicing. this often does have a tissue specificity to it, and we often refer to the different products, the protein products that are produced from isotherms, from alternative splicing, as isoforms. So we refer to them as isoforms of different genes or isoforms of a particular protein. And isoforms are then produced off the same gene, but through different combinations of exon utilization through alternative splicing. So to summarize, alternative splicing is the phenomena by which genes for primary transcripts may be spliced in different ways. And that can be through various forms of intron retention. It can be through exon skipping. It can be through alternative splice donor acceptor sites and that lead to alternative exon utilization. And at the tissue-specific level, we can have what are said to be tissue-specific isoforms. And we have some examples of that in nature. We're going to be talking about the WT1 protein, the CYT1 protein, and the CDKN2A protein. So there are many, many examples throughout nature, but in your textbook, we're going to specifically focus on these three examples. So this picture shows on the top, we have the alternative splicing of the WNT1 gene, and on the bottom we have the alternative splicing of the ERBB4 protein. And what you can see in the WT1 protein, and that's sometimes referred to as the Wilms tumor protein, because it plays a role in Wilms tumors. You can see due to alternative splicing, you may have a particular form of the protein that has either 17 more or 17 less amino acids, and depending on how it is spliced, that the two isoforms may behave differently in the cell. And in the case of the WT1 protein, the minus KTS version of this protein acts as a general transcription factor, whereas the plus KTS form does not. In the ERB4 protein, again, we have some alternative splicing that can lead to different numbers of amino acids, because obviously if you include or don't include a particular sequence of RNA, that's going to affect the actual length of the polypeptide that's translated. But be that as it may, they have different roles in the cell, and one functions as a tyrosine kinase, and one does not. So this is very important. One acts in signaling, and one does not, because they lack a binding site for the phosphatidylinositol 3 kinase signaling molecule. So depending on which, the key point in this slide is that these examples show how alternative splicing leads to different lengths of the polypeptide that's produced, and then in turn that alters the function. So they may have a different function. The alternatively spliced forms may have different functions, and that's what's shown in this slide. In our next example, we actually show how alternative splicing of the CDKN2A gene produces two entirely different tumor suppressor proteins. In one instance, one way of splicing produces the P16 INK4A protein, and in another form of splicing, we have the P14 ARF protein. Both of these proteins are involved in cell cycle control. And you can see here from this slide that it really it depends on how the exons are joined together. So we have for the P14 ARF, we have the 1 beta joining with exon number 2, and for the P16, we have the 1 alpha joining with exon 2. And it's pretty clear from the diagram how these two are pieced together, but one gene, different proteins. Another example of altering RNA that has already been transcribed is through RNA editing. And etologically, this is the result of deamination or transamination. And it's a natural process. It's not well understood, but at a functional level, deamination of the adenosine nucleotide, for example, leads to the production of inosine. And why is that important? Because if you have an A being switched out for an I, that actually changes the base pairing. So now you have a CAG for GCG, and then that it could lead to a change in the amino acid that's actually there, a glutamine being switched out for arginine. So if we change amino acids, particularly those kinds of substitutions, they are fairly significant. So now let's talk about regulation of gene expression at the level of translation. So just to recap, we've talked about regulation of gene expression at the transcriptional level. So if we don't transcribe the RNA or we influence how much RNA is produced, we're regulating gene expression. We've talked about it at the splicing level and RNA editing level, how we splice or edit the primary transcript will also influence what protein is actually produced. And now we're going to talk about how the regulation of translation influences gene expression. So we can have transacting regulatory proteins that influence translation. And regulation at the level of translation allows cells to respond more rapidly to altered environmental stimuli than is possible by altering transcription. So transcription takes time. Transcribing the mRNA, making that primary transcript, and then splicing it, that takes time. So if there's some way to sequester already transcribed and processed, that means spliced RNA, and then you'll be ahead of the game. So as an example of that, we're going to look at iron metabolism and the iron responsive elements. So this picture shows an iron responsive element, and actually our single-stranded RNA, which RNA is single-stranded, only DNA is double-stranded, it can actually adopt a variety of secondary structures as is shown here. And these secondary structures help to extend the lifetime of the RNA and protect it from degradation. So normally RNA, if it's just in a linear form, it's more susceptible to degradation. And so in this slide we see what's known as the iron responsive element in human ferritin mRNA. And ferritin is actually a protein that's involved in the levels of iron in the body. So ferritin is an iron-binding protein that's used to store iron in cells. And it works synergistically with transferrin, which is a receptor that helps us to absorb iron from the diet. So if we have lots of iron in our body, then ferritin, which is an iron-binding protein, will help to sequester that iron and store it in the cells. But if we have low iron in the cells, then the protein transferrin, which is a receptor, will help us absorb iron from our diet. And so when iron levels are low, the priority is to maximize the amount of iron that can be absorbed from the diet. and the transferrin receptor mRNA is protected from degradation so that it can make a protein product. Conversely, when iron levels are high, ferritin production is activated to store iron in the cells. This actually will happen without any change in the production of ferritin or transferrin receptor mRNA. Instead, both of these mRNAs have these iron-responsive elements, which can be bound by specific iron-responsive element binding proteins that regulate the production of protein from these mRNAs. So these mRNAs are in the cell, they're ready to go, they're in this hairpin structure, and then depending which iron-responsive element binding proteins are present, which in turn depends on the iron levels in the cell, we will express either ferritin or transferrin receptor. And that's what's shown by the picture here in this slide. When iron levels are low, a specific IRE binding protein is activated and binds the IRE in the ferritin heavy chain gene, and also IREs in the 3' untranslated region of the transferrin receptor. Binding inhibits the translation of ferritin, but protects the transferrin receptor mRNA from degradation, maximizing the production of transferrin receptor. When iron levels are high, the IRE binding protein is inactivated and maximizes the production of ferritin and decreases the production of transferrin receptor. So it's a very nice synergistic process. So next, let's talk about post-transcriptional gene silencing by microRNA. So again, transcription has taken place, the gene splicing, or the RNA splicing has taken place, and now we're regulating the actual translation of this RNA. The transacting regulators that we just spoke about in the iron-responsive elements used to be viewed as unusual exceptions. But the discovery of RNA interference and the existence of tiny RNA regulators, specifically microRNA, changed all of that. And so in this final section of this module, we'll be talking about RNA interference, or RNAi. We'll be talking about SI, or silencing RNA. We'll be talking about how we can repress the repressors, and finally, circular RNA. RNA interference has actually been something that underlies natural cell defense mechanisms. For example, viral and transposon RNA can be inactivated using RNA interference. In this slide, we see that there is an enzyme in animal cells naturally occurring. Long double-stranded RNA is cleaved asymmetrically by an endoribonuclease called dicer that's specific for generating double-stranded RNA, and these are those 20 to 20 nucleotide pieces that we talked about in the previous slide with overhanging 3' ends that are called short interfering, or Si RNA. And the Si RNAs are bound by protein complexes that help to cleave and generate these fragments. And then in turn, with other proteins, these fragments hybridize and recognize specific RNA and lead to the degradation of particular RNA, thereby eliminating, in the case of like viruses, for example, the foreign RNA sequences in the cells. This slide depicts how microRNAs are produced and work in cells. In the left side here, we have miRNA genes that are transcribed and cleaved in the nucleus to generate a stem loop RNA that is then subsequently exported into the cytoplasm and gets further cleaved by endoribonucleases known as dicers to generate an mRNA duplex. which is then cleaved and degraded, leaving one other strand, the guide strand, as a mature single-stranded miRNA. So these mature miRNAs are single-stranded, and they're shown on the right-hand side of this picture in brown. And you see that they're binding to specific loci where they do so through base pairing. And shown here are, just for illustrative purposes, the miRNA that was produced in the cell for 5 different genes, depicted A through E. And finally, as we have begun to understand more in depth the role of miRNA, we have how do we get rid of the miRNA? You know, this is being produced in the cell. We see that the miRNA is regulating gene expression, and so repression of the repressors, competing endogenous RNAs that sequester the miRNA. Now, so we're actually having something that can bind to and influence the miRNA, and that's sometimes comes from pseudogenes, and pseudogenes are genes that don't have any functional gene product produced, but they're transcribed. So many of our pseudogenes are known to be transcribed, and some of them seem to have undergone purifying selection. That is, that they persist throughout evolution. And a landmark study that was published in 2010 provided the first insights into how functional pseudogenes worked and showed that the human PTEN gene is regulated by a highly related processed pseudogene, PTEN-P1, that is located on a chromosome adjacent to that which the PTEN gene is made from. And so that's very interesting. The PTEN-P1 protein encodes a non-functional RNA that regulates the P10 miRNA regulators. In addition to material produced off of a pseudogene, there are circular RNAs, and these circular RNAs are produced through the process of head-to-tail splicing, and they function as miRNA sponges. And they're found ubiquitously, and they're very abundant, for example, in human fibroblasts. And this slide actually just depicts the P10 miRNA binding sites, which in turn influence the regulation of this gene by the microRNA binding. And lastly, we show how the circular RNA can be used as a sponge to bind up the microRNA, and that in turn regulates the regulator. And this concludes the first module of Chapter 6. So microRNAs and RNA interference. miRNA, which is short for microRNA, is an ssRNA that can downregulate the expression of specific target genes. Typically, miRNA are 20 to 22 nucleotides in length, and they're formed through multiple processing events. So this is produced in the cell through multiple processing events. The miRNA actually binds to complementary regions, so that means by nucleotide base pairing, and they're very important in gene silencing. Overall, the mechanism by how they silence genes is not well understood. It's not certain whether they enhance RNA degradation or they physically inhibit translation event itself or a combination thereof. Up to this point, we've talked about regulation of transcription as a means of regulating gene expression. So what about once the RNA has been transcribed? What about things that influence RNA processing? Because as we've learned in previous slides, that first the RNA is transcribed into the primary mRNA transcript, and after that it's processed prior to translation. So we know that genes contain introns and exons, and the primary transcript will still have the introns, and these need to be spliced out. So RNA splicing is an important aspect of genetic regulation in RNA processing. And it has a couple important DNA sequences, again, that are critical to the splicing event, and those are the splice donor and splice acceptor sites. So we're going to actually cut out an intron, and so we have to have something that demarcates where the intron begins and where the intron ends, so that the splicing machinery of the cell can know how to do that. And also, we have to consider the other aspect of splicing, which are exon skipping and isoforms. So these all of these things play a role under the general banner of RNA splicing. And lastly, there is RNA editing. So these elements are what we think of when we talk about genetic regulation in RNA processing.